Today I'm going to present my work about synapse development. The big question our lab is interested in is how cytoskeleton regulates synapse formation, and the specific protein I work on is actin. And we know human brain is an extremely complex system with hundreds of neurons connected with each other. A typical information flow within neurons then drives that information from
Can you turn on the mic?
Everyone can be ready? Okay, so the typical information flow within a neuron then drives collect information from the receptor organ or other neurons, and then those information will be integrated in the cell body, which will be passed on to axon to another neuron. Between the axonal terminal and another neuron, there is a special connection point that is synapse. Synapse is the primary site for neuronal communication. These three are axonal terminals. As you can see, synapse can be formed on cell body or dendrites. For most excitatory synapses, they reside on the tiny protrusions from dendrite, named the dendritic spine. This is a dendrite from primary capsule hippocampal neuron, and those yellow tiny protrusions are dendritic spines. In this image, only the postsynaptic part, which is dendritic spine, is labeled. All of them are innervated by one or multiple presynaptic terminals, which are not fluorescent labeled, so you cannot see them here. In this image, most of the spines are mature. They exhibit mushroom-shaped structure that's an expanded spine head with a very thin neck. During development, there's a precursor form of dendritic spine, which is named the phallopodia. Phallopodia are thin and long protrusions without distinguishable head. They explore around the environment until they find the axon terminal and make the contacts. After making the contacts, they gradually transform to an expanded spine head together with postsynaptic components clustering in it. We care about the process of spine development. That is because the alteration of normal spine development or maintenance is associated with neurological disorders. If during development, if the phallopodia initiation or the transformation to spine is disrupted, there will be problems of neuronal connection. One example is in fragile X. These three are dendrites from postmortem brain. The left two are from normal subjects, and the right one is from fragile X sample. So we can see the spines in fragile X, they are thinner, longer, with smaller heads. And also the density appears to be higher. The abnormal spine formation in fragile X is prominent, and it is considered at least partially responsible for the neuronal defects. There's another type of spine alteration, that is spine loss and shrinkage, which is associated with neurodegeneration, such as Alzheimer's disease. In the animal model of Alzheimer's, the animal studies have revealed that spine loss happens way ahead of neuronal death, and this could be underlying the early cognitive decline. So we know that spine alteration is associated with brain disorders. If you have some problem of spine structure, quite likely there will be some problem of neuronal connection. So what regulates spine structure? The main structural support for spine is actin cytoskeleton. This is an electron microscope image showing actin filaments within the spine. The spine is pseudo-colored blue here. As we can see, actin filaments are enriched in spines. They form a bundled structure in the neck and stay highly branched in the head. So this highly branched network not only provides an overall structural support, they also participate in many neuronal specific activities, for example, organization of the postsynaptic density and the receptor trafficking. To well accommodate those reorganizations happening in the spine, there are many acting regulatory molecules concentrated in the spine as well. One of them is actin. So actin protein, also named as CAPC, it binds to the growing end of actin filament and prevents actin polymerization. Once you remove capping protein, the actin filaments will keep polymerizing and growing fast. One example is if you cut your finger, then the platelet is activated. There will be a molecule to remove capping protein from the actin filaments. Then those actin filaments in the platelet will grow really fast to spread out. Eventually, they will form a blood clot together with other substances. Capping protein function by forming a heterodimer of alpha and beta subunit. Beta 2 is a predominant one in the brain. Later in my slides, you will see most of my manipulation is on the beta 2 subunit. So what we know about capping protein in the brain? First, we know it is most abundant in hippocampus, as shown by the situ data. Also, we know it is present in dendritic spines. Again, this is an electron microscope image showing spine structure labeled blue here, and those gold dots represent the staining of capping protein. You can see capping protein are quite abundant in the spine head. Based on this previous finding, we decided to use hippocampal neuron as a model to study capping protein function in dendritic spine. Although we don't have much information about capping protein synapse yet, there are sporadic reports about capping protein level was found to be altered in Down syndrome, dementia, or Alzheimer's. This indicates capping protein may be important for cognitive function, and we think our study regarding capping protein in synapse development may help elucidate some mechanisms on the cognitive impairment in the future. So our main hypothesis is capping protein is an important regulator of spine formation. The first question we want to make sure is it expressed at the time of spine formation? If the answer is yes, we can continue to examine the function. Is it capping protein required for proper morphology and density of dendritic spine? Is it required for normal synaptic transmission? So the first question, is it expressed during the time of spine formation? We performed Western blot to examine capping protein level at different developmental stages. Black babies are usually delivered on embryonic 22 days. And the first 2 postnatal week from P1 to P14 is the peak time for spine formation. We chose 3 time points here, embryonic 18, P9, and P18, which roughly corresponds to before, during, and after peak spine formation. We sacrificed the 3 animals for each time point and performed the Western blot with a hippocampal assay. We see both alpha and beta subunits of capping protein exhibit an increasing trend of expression level. And if we look at P9, which is during the peak time of spine formation, there's an elevated capping protein level, which is maintained at P18. So basically, this Western blot tells us it's indeed expressed at the peak time of spine formation. Although it's expressed at the time, it doesn't necessarily localize to where we want to study. So we do perform the analysis to examine subcellular distribution of capping protein in neurons. The GLP tagged the capping protein together with M-orange were transfected into neurons. You can see the for this M-orange, which is uniformly distributed, the capping protein is preferentially localized into dendritic spines. If we analyze the ratio of capping protein versus M-orange, we can see those hot spots, which represents high ratio, that is accumulation of capping protein in dendritic spines. So from Western blot data, we know it is expressed during spine formation, and now we see that they are accumulated in spines. We can continue to examine their capping protein function now. The question we asked is it required for proper spine morphology and density? We use loss of function approach. So 3 groups of neurons are used. The control group, we basically just transfer neurons with GLP to illuminate morphology. Then the second group is knockdown group. We have a short hair spin ion specifically targeting capping protein, and also a GFP to illuminate neuronal morphology. The third group is knockdown rescue group. That is a shRNA specifically targeting capping protein together with an encoding sequence of capping protein. That coding sequence is refractory to this shRNA. So this way we can examine if the knockdown effect can be rescued by reintroducing capping protein back into the neuron. If indeed rescued, that means our knockdown phenotype is specifically due to capping protein, not due to some off-target effect. We use the primary culture hippocampal neurons. In vitro system, the peak time for spine formation is DRV14 to DRV21. So we transfected neurons right before DRV14 and performed imaging on DRV21 to examine spine morphology. What we see here is this neuron is from control, this one from knockdown, and this is from knockdown rescue. The bottom row are just 3D reconstructions of those images. In control, we can see most spines are mushroom-shaped. They have a big round head and a relatively thin neck. But for knockdown group, lots of them are still seems like a phalangodia shape. They don't have a round head. Some of them are branching. And for the others, like this guy, it has a round head. There are tiny structures coming out from the head. This aberrant morphology is not observed in the knockdown rescue group. So to better depict the differences, we use the 3D reconstruction to see the spine morphology. In control, most of them, they are round, they have a round head, smooth, but for the knockdown, lots of them are this phalangodia shape. They might be branched. Even if they have a round head, there are protrusions coming out from the head. This may be even better depicted with the normal spines are like closed fist. Then you got pointed finger, victory, or half open arm from the knockdown. So I'll summarize my data here. It's amazing, almost for every abnormal morphology, I can find a magic. Morphology and density. So now we have data showing that the morphology is disrupted. We want to know if the density, we want to do some quantification about spine density. We use the simple criteria here, which is also widely used in other literature. Just the spine are defined as those structures with a spineless head, as a distinguishable head. If you have protrusions like this without distinguishable head, we quantify as phalangodia. So this is the result. GMP group, which is our control, for each 100 macron dendrite, we get about 60 spines together with very small number of phalangodia. But for knockdown group, we get about 30 spines together with another 30 phalangodia. The knockdown rescue group is closer to control. If we go back to our model of spine development, phalangodia arise from dendrite. Upon contact with axonal terminal, they gradually transform to spines with a head. So we see a drop of spine density accompanied by increased phalangodia density. We think this may suggest that capping protein is important for the conversion of phalangodia to spine. I will summarize my second part of data here, that is capping protein knockdown will disrupt spine morphology and reduce spine density. What we do want to know is whether functionally the properties of synapse are indeed affected. So to analyze synaptic transmission, we first did some staining with functional synapse markers. That's presynaptic marker SV2 and postsynaptic PS95. So we quantify whether the functional synapse density is also affected. All the green signals you see here are just the GFP to label dendritic spine. So that's all the GFP. And for presynaptic terminal marker SV2, if there is a functional synapse formed, you should see SV2 signal that's red here opposed to the spine, like this one. And for the postsynaptic marker PS95, you should see it within the spine, that is like within those protrusions. So after merge, you should see yellow. If we look at the density of those red contrasts here, for both SV2 and PS95, after capping protein knockdown, the density is lowered. And we also have quantification, which is not shown here. Basically, it says capping protein knockdown reduces the density of functional synapses. We further, so the golden criteria, if we are talking about synaptic transmission, is electrophysiology. So we collaborated with some electrophysiologists to perform whole cell patch clamp to record the miniature EPSCs. The frequency of miniature EPSCs can be considered as a reflection of the functional synapse number in the neuron. Again, we can see a significant decrease of MEPSC frequency after capping protein knockdown. And this capping protein knockdown rescue group can also rescue the physiology here. So the answer to the question, is capping protein required for normal synaptic transmission? We think is yes, because baseline synaptic transmission is impaired after capping protein knockdown. So I'll summarize my data here. Capping protein expression increases at the peak time of spine formation and accumulates in dendritic spines. The knockdown of capping protein impairs spine formation and morphogenesis, as shown by aberrant morphology and decline in spine density. Also, baseline synaptic transmission is reduced after the knockdown. So how is capping protein involved in this process? We put what we think it is doing there in a hypothetical model. Although we haven't proved it yet, this model can explain what we observed. So if this is an axonal terminal, and you have a dendrite here, phylopodia will arise, trying to grow and contact the axonal terminal. It keep growing. Those long filaments within the spine, these guys, within the phylopodia, they keep polymerizing until you have capping protein to stop them. The reason you want those filaments to be stopped is because if you want the head to expand, you would prefer those actin monomers to be added on the side branches instead of keep elongating. So then there can be an expansion of the spine head and more capping protein coming to keep those branches short and keep branching out. And at this time, with the branched network, the post-synaptic density can be supported and formed. Eventually, they end up with this perfect mushroom-shaped spine. That's what we see here. How about in the knockdown neuron? This is axonal terminal, dendrite. The phylopodia keep growing, but at this time, there's no capping protein to stop those long filaments. So monomers, actin monomers, cannot be sufficiently added to those side branches. It will keep growing, then you end up with this phylopodia-shaped protrusion. And if there happens to be one side branches really grow really low, you may end up like this. We do know that our knockdown is not 100%, so there are some protrusions. They will still be able to grab some capping protein. So in this case, I think they grow here, they grab some capping protein, and they were able to make the transition. Eventually, they also form post-synaptic density well. However, since this is knocked down, so capping protein level may be still lower than others. So it may not be sufficient to restrict all the filaments there. So there will be some actin filaments protruding out, and so you see this morphology, like this guy. So I think this is our hypothetical model. And the take-home message here is capping protein is an important actin regulatory protein for spine formation and the synaptic transmission. I'll stop here and acknowledge my mentor, Dr. Zheng, and all the members in my lab. Thanks for their support and helpful discussion. I also want to thank Dr. Chris Hassel, Pai Gongchen, and Xin Kang for electrophysiology, Dr. John Hammer for reagents. Besides that, I want to thank from Imaging Core Facility for help with image analysis and 3D reconstruction. At this time, I want to do a free ad for the Imaging Core Facility for those candidates who are really interested in quantitative imaging. Emory is has a wonderful resource. In the core facility, we have a large collection of fancy microscopes and support from the very knowledgeable staff members. In the end, I want to thank my dissertation committee and the neuroscience program. It is great experience to be on hands of faculty members who really care and to be surrounded by a group of students who are really supportive and accomplished. Thank you very much.